eukaryotes is mediated by, it is mediated by, okay. So intracellular communication in eukaryotes is mediated by. So this was the question here. So in this question, what we can see is, so A is a lysosome. So this term is lysosome. But you know, lysosome is not correct answer. Lysosomes are usually what are present there. In lysosomes, there are presence of hydrolytic enzymes. Hydrolytic enzymes are there. So these enzymes are related to destruction of carbohydrates, proteins, amino acids, as well as lipids. Okay. So there is no communication between two cells in lysosome. Then there is this exosome. So this is the correct answer here. Exosome. So exosomes are the particles which visit a cell. So exosomes are the particles which will invaginate here, and through this invagination, these exosomal particle will go out. Okay, so they will go out, and so they will act as communicating material, communications. So these communication material are nothing but exosomes. So the word exo means outside, something which is going outside. Third choice was peroxisomes. So peroxisomes are the sites where usually oxidation reaction happens. One such example could be very long chain fatty acid oxidation. So VLCFA oxidation happens in peroxisome, and then there are peroxisomal storage disorders like diseases comes in this category. So MCQs have also come from that part, this VLCFA. Diseases example could be Zellweger syndrome, Zellweger. Okay, so Zellweger syndrome. Such syndromes are peroxisomal type. Then what are these microsomes? Microsomes are nothing but these are endoplasmic reticulum particles. Okay, endoplasmic reticulum particles. So these microsomes, why they are called microsomes? Because like exosomes, they are small particles like that, spherical particles. Usually ER is what? Elongated particles. These elongated particles are converted into these small spherical particles. How? Using a technique known as ultracentrifuge. Ultracentrifuge. So small particles, since they are in micro shapes, small shapes, so they would be known as microsomes. So the answer here is intracellular communication. Answer is exosomes. Okay guys, so this is the first question which came this 2020. We move on to the second question, both glycolysis and gluconeogenesis. So this we have to see, glycolysis and gluconeogenesis. So what is this? Both the enzymes, actually in glycolysis there are some rate limiting steps, and those steps are irreversible steps. So if I write glycolysis on this side and gluconeo on this side to compare the rate limiting enzyme, so these could be hexokinase this side. The opposite of hexokinase here is glucose 6-phosphatase. Second enzyme is PFK1. Here, opposite of this is fructose 1,6-bisphosphatase. Third example is pyruvate kinase. Pyruvate kinase, and this pyruvate kinase here is Two enzymes are there, PC, pyruvate carboxylase, and PEPCK. So phosphoenol pyruvate carboxykinase. So these are the equivalent enzymes in both these places. Okay, these two things will come equivalent to pyruvate kinase. Now question asked is enzyme present in both glycolysis and gluconeogenesis. So what do you mean by this? Both glycolysis gluconeo. See, what is this? PFK. PFK is found only in glycolysis. PEPCK only found in gluconeo. Pyruvate kinase only found in glycolysis. So only one choice remains, C choice, and this C is the answer here. So C states that phosphoglycerate kinase is the enzyme which is present both in glycolysis and gluconeogenesis. Okay, so the answer becomes C. Third question, which of the following would be incorrect in RH incompatibility? So this they are asking RH incompatibility. So what is happening in RH incompatibility is there is presence of hemolysis. So hemolysis is present here in RH incompatibility, and in this hemolysis what is going to happen is this will lead to increased level of unconjugated bilirubin. Increase in unconjugated level of bilirubin, and this unconjugated bilirubin, then see the first choice, raised unconjugated bilirubin. Yes, this is true, and this unconjugated bilirubin, part of this partly conjugated, actually some part is conjugated, and this conjugated stuff then will go into the colon and then from there into the stools. So in the stool, urobilinogen, which is the converted form of bilirubin, is excreted out, and through enterohepatic circulation, what will happen? This will be into the urines as well as. So urine, urobilinogen. So this will also be raised. Everything would be raised, but directly bilirubin is not excreted. This is not excreted because reason, unconjugated bilirubin is bound with albumin, and this combination albumin and unconjugated bilirubin can't be so what is there? There is bound with albumin, and this albumin can't be filtered out into the kidney. So if it is not filtered out, it is not in urine. So this bilirubin is not found in urine. So this is correct. Raised stool, yes. Raised urine, yes. But increased urine bilirubin, no. So this is incorrect. So the answer is here D. Which of the following is incorrect? So this is the incorrect choice. 3 ka answer is D actually. Okay. Fourth, vitamin deficiency causing neonatal seizures. So here is the question, which vitamin is related to seizures? Actually the answer here is vitamin B6. B6 is related to these different types of seizure activity. So the answer is, B6 is what? Pyridoxine, B6. Okay. So this becomes the answer here. So why it causes seizures? This is one question. So seizure activity is due to decreased GABA. And you know that It is an inhibitory neurotransmitter GABA, and when it is decreased, the brain activity will increase. So that will cause seizure type of action. So why GABA is decreased? Because B6 is involved in a set of reactions known as decarboxylations. Decarboxylations. And these decarboxylation reaction, one of them is glutamate is converted to GABA when you remove CO2 from it. So this is a decarboxylation and this is B6 dependent. So if no decarboxylation, no GABA. No GABA, seizures, neonatal seizures. So from curiosity's sake, can you tell about thiamine? Thiamine is B1, flavin is B2, pentothenate is B5. So some diseases also comes in this category. B1 deficiency क्या होती है? This is beriberi. B2 deficiency, glossitis, chiliosis, etc. Cracking at the angles of mouth, such things are seen in B2. B5 deficiency, this is Gopalan's burning foot syndrome. Gopalan burning foot syndrome. So these are examples of different vitamin deficiencies. So here the answer is the fourth one. Okay, so let us move on to the next one, fifth question. Calorie from TPN, total parenteral nutrition. So such nutrition is given to the patients who cannot eat from mouth due to some surgery or due to their chronic bedridden condition, in a comatose condition. So you have to give totally in a parenteral form, means IV आपको देना पड़ता है. So you will give those different carbohydrates, amino acid, lipids, vitamin, etc. all through directly IV route. So now they are asking, if you are taking this TPN and we are giving this diet, how much calorie will this person will get? So dextrose here is nothing but glucose. Then we have given amino acid. Then we have given lipids. Now you might be knowing from glucose how much energy is formed. So around 4 joules. So around 4 joules per milligram, like that is energy is released. Amino acids same 4, but lipids are around 9. Okay, then we will calculate here 100 into 4 plus there is 30 amino acid, so 30 into 4 plus lipids are 9, so 40 into 9. So this is 360 and 120 and 400. Like that you can see. So if you sum total them, this is 760 and around this is coming total is around 880. So 880 joules we will get. So the answer is D, 880. Okay. I hope this is clear to you. Now, next question which came was, which of the following is not an enzyme in cholesterol biosynthesis? So, simple, 3-4 steps you have to see in cholesterol biosynthesis. The precursor molecules is acetyl coenzyme A, acetyl CoA. 2 molecules of acetyl CoA combines to form acetoacetyl CoA. This forms HMG CoA, and finally this forms a compound mevalonate. So 3 steps are there, 3 enzymes, and we should know about these 3 enzymes first. Mevalonate finally converts to cholesterol by various steps. So what are the enzymes? thiolase comes here. So this is the thiolase. Next is HMG-CoA synthase. You are synthesizing. So synthase or synthetase, this will come here. Then HMG-CoA to mevalonate. This is a reductase enzyme. HMG-CoA reductase. So reductase. So what is remaining? Lyase. So this is not N, this is HMG. HMG. HMG-CoA lyase. So this is the answer. So my question to you is, where this enzyme is found? This, if you lyse this HMG-CoA lyase enzyme, this HMG-CoA will convert to what? This will convert to acetoacetate, which is nothing but a ketone body. So this is the enzyme in ketone body synthesis. In ketone body synthesis, this enzyme is found. Okay, so this is about cholesterol biosynthesis. So these are 6 questions. Let's go to the 7th. Correct about the vacutainer. Now what are vacutainers? Vacutainers are the syringes, or these are the containers which will collect the blood. Now blood may be collected in EDTA. So all the hematological exercises are done in EDTA because this has got unclotted blood. Or they may, maximum biochemistry samples are happening in the serum. So serum is for the whole biochemistry reactions, biochemical test. Okay. So there they are asking, correct about given vacutainer is you can see the small part serum is written here, serum. So the serum means this serum biochemical. So how do you form serum from this blood? By a clotting mechanism. You know what is serum? Serum is clotted blood. So the cellular part you remove, whatever clotted the fluid part remain, that is serum. So the answer here becomes clot activator. Sodium fluoride, where it is used? Blood glucose estimation. Blood glucose estimation. So in blood glucose you have to use sodium fluoride, because one question comes, why? Because they inhibit a enzyme enolase. This is also one of the MCQ. Okay. Now heparinized serum. Heparinized serum is used in something like acid base ABG, arterial blood gas analysis, in which heparinized sample is used for that purpose. So various purposes I have shown you, where which vacutainer. So what you have to see? You have to see which serum is written, or EDTA is written, or NaF sodium fluoride is written. So you have to see for the clue in the diagram. Okay. Now vitamin D synthesis sequence. So this thing starts from skin actually. 7-dehydrocholesterol. 7-dehydrocholesterol in the skin converts to cholecalciferol, CCF. This CCF converts to 25-hydroxy cholecalciferol. Another name is calcifidiol. So one more all is attached here, 125-dihydroxy cholecalciferol. This is calcitriol. Calcitriol. So these are different stuffs we have seen here, and they are done by enzymes. Here UV light. Here 25 alpha hydroxylase. and here 1 alpha hydroxylase. Now this is the rate limiting step, and this is the kidney step, this is the liver step, this is the skin step. So what is the order? From skin to liver, from liver to kidney. So which is the correct answer? A. Skin to liver and from liver to kidney. Okay. So this is about the 8th question. 8th question. 9th question. Glucose is stored in glycogen form Y. Okay. It is seen in glycogen form Y. So compact, yes, this is a true statement. Glycogen is a compact thing and they are having spherical structure. Actually, glycogen is spherical, which consists of multiple small glucose like that inside it. So around 1 million units in one glycogen unit means glucose units. So this have got a small surface area as compared to individual 1 million glucose units, small surface area. So they will cause less surface tension. So they will occupy less volume. So they are more compact. So the A choice is the most appropriate one. Second choice, can be reduced from multiple branches end. So they can be reduced from. No, multiple branches they cannot be reduced from. Means there are multiple branches. They are actually reduced from one branch. They can be stored at multiple sites. At only 2 sites they are stored. One is your liver, other is your muscle, and not multiple sites. Then can provide glucose as much as needed and when before 1 week. Not as much as needed, only the stored amount of glucose would be the thing which you will get. And when needed before 1 week, they are saying, not 1 week. Actually, glycogen will give you max till 24 hours of fasting, not 1 week. Okay. So the D choice is also wrong. So best choice here is A choice, compact. Let's move. DNA methylation is not related to. Now this methylation word means this causes transcription inhibition. Inhibition of transcription. This is seen in DNA methylation. So it is not related with. This is they are asking. So one by one, if you see, D choice will go capping. The word capping is related to mRNA. So if you see mRNA, there is a cap, there is a tail. This cap is made up of 7-methyl guanosine. So this is methyl. That's why this is correct. Capping has methyl. Gene silencing, yes. Transcription inhibition. So gene को silence कर देता है, transcription नहीं होने देता. Gene silence, it causes gene silencing. Then comes mismatch repair. Yes, mismatch repair is a type of repair where mismatch repair. Here the repair mechanism, defect is due to copying error. Means during copying from one copy to another when it is formed, this is a mismatch error. So this error is removed with the help of this methylation. Okay. So how do you methylate? Like one example, if I tell you, suppose this is a GATC, this is the daughter strand. So G pairs with C, this is okay, but in place of C, let's say it is A. So this is the mismatch. So you have to replace it with C actually. So how do you replace? By first putting a methyl group here. So when you methylate, this methylation is at the parent strand. To identify which is the parent strand, you methylate. Add adenine residues. Adenine residues. So what do you mean by this? This is a question which comes. So here it is actually A, adenine. So this A is adenine residue. This is activated. This methylation is done. And then we know that this is the parent strand. And then C should be put here. If you do not know this methyl, you can do opposite also. In place of this G, you can put a T here. So to remove that, you have to do what? You have to methylate the parent strand. So this is also good. So what is the answer? Answer is B, DNA replication. It is not related to DNA replication. So this we have seen 10 questions. This is the 11th question which came. Which value not required to measure creatinine clearance? So what is creatinine clearance? So this is volume of serum cleared of a substance per unit time, that is per minute actually. So in 1 minute, how much serum is cleared of a particular substance? This particular substance is nothing but creatinine. So this, if I write creatinine clearance, CR clearance, this is equal to UV by P. UV by P. So this U is urinary concentration of creatinine. V is volume of urine. P is plasma concentration of creatinine. So this is actually a physiology question also, you can say. You can put it anywhere, biochemistry or physiology. So UV, volume and concentration you are multiplying. So what is coming? Volume और जब concentration को आप multiply कर देते हैं तो amount आ जाता है. Here also it is amount. Amount of creatinine in plasma is equal to amount of creatinine in the urine. So from this it is derived actually. Whatever creatinine amount is there, that amount is going into the urine. This is the hypothesis. So amount of creatinine in plasma would be what? Simple thing. Plasma concentration, this one, plasma concentration into the volume inside the volume of plasma, creatinine clearance. And this should be equal to U into V. This is U and this is V. So this is derived from this formula actually. UV by P. Now question asked is, which value is not required to measure? So urinary creatinine, yes it is needed. Serum creatinine, yes it is needed. 24-hour urine volume, yes. Volume of serum passing through kidney, no, this you cannot assess. And this is actually what is creatinine clearance. So C is the answer of choice here, which is not required. Okay, 12th question. Dicomerol. So this dicomerol is actually a inhibitor of vitamin K. So this inhibition causes what? Bleeding tendencies. Okay, bleeding tendencies. So how it causes bleeding tendency? You know, vitamin K causes gamma carboxylation of glutamate residues of factor 2, 7, 9 and 10. 2, 7, 9, 10. Gamma carboxylation. Now these gamma carboxylations, how this happens, you can see here. Let's say this is any factor. Factor is clotting factor, which is a protein having a glutamate residue. This glutamate is converted to gamma carboxyglutamate. By what enzyme? Carboxylase enzyme. This carboxylase is vitamin K dependent. This carboxylase also known as epoxidase. This epoxide causes one hydroquinone derivative of vitamin K. Vitamin K has got a hydroquinone derivative. From this actually epoxide derivative of vitamin K forms. Epoxide is converted to quinone derivative. This to this, this to this. So this is how this vitamin K is helping. Here it is done by epoxide reductase, which is inhibited by our this dicomerol. Also warfarin. So warfarin and dicomerol acts here. Okay. So dicomerol inhibits vitamin K by. So this is an example of competitive inhibition. Competitive होते हैं. तो जैसे ही आप you add more vitamin K, dicomerol inhibition is lost. Okay. So this is an example of competitive inhibition. So 13th question, ammonia is detoxified in brain. So ammonia is toxic. It is toxic to brain. So because of this toxicity, you have to trap it. Trap the ammonia. So how it is trapped? Trapping is done with the help of glutamate residues. So glutamate trap ammonia converts to glutamine. This glutamine goes to liver, where from this ammonia is extracted to urea, and this urea is finally excreted into the urine. So this is the usual way. So in brain detoxification, answer is glutamine. So you can write one more point. All tissues in the body, ammonia is carried by glutamine except muscles. In the muscles, the carrier is actually alanine. Alanine is the carrier. Okay, so both comes as a MCQ. So two are there. Here the best answer is glutamine. Alanine would be an answer in muscles. Urea and arginine, no, they are not good choices. This is 13th. 14th, identify the image given below. So you can see two molecules are there. This is an example of GAG. One molecule, two are there. These two are attached to each other by some molecules. This is the carbon numbering 1, 2, 3, 4, 5 and 6. At the 6th position there is a carboxyl group. So the first is actually glucuronic acid plus, which is the second one, NAC, N-acetyl and GOH, glucosamine, N-acetyl glucosamine. And this combination is found where? This combination found in chondroitin sulfate. So the answer is the last one, chondroitin sulfate. So what are the other containing molecule, can you tell? Keratin sulfate, what this is consisting of? Keratin is galactose plus NAG. Dermatan sulfate. Dermatan is iduronic acid plus NA-gal. Heparin. Heparin is iduronic acid plus glucosamine. The last answer here is chondroitin sulfate. So little bit about structure we should also know here. Okay. So this many question came actually in this AIIMS exam 2020. So it was more or less easy questions. So little bit of concept you have to draw, and easily you can solve these MCQs. Okay guys. So with this we have finished this different types of discussion here, and with this we will finish the discussion. Okay. Thank you.